Thanks to the organisers for the invitation to come and speak. I've really enjoyed the meeting so far. So we've had a great introduction to stem cells and pluripotency already, and we've also had a lovely introduction, thanks to Nathan, on transcripts and transcriptional profiling. And I wanted to open my talk by just reminding you that tissues and cells can be grouped or clustered by the networks of genes that they express. So Nathan's quite right, transcriptomics is an old method. It's still a workhorse because it is the single technology where we can gather the most information from a single population in one hit. And we can't do that, unfortunately, at the moment for proteins, and we certainly can't do it for metabolites. And what you're seeing here is simply every dot represents a different cell or a different tissue type in an atlas. This is the Phantom 5 atlas, which was published a few years ago now. And the second point I want to make from this slide is the fact that when you profile cells in isolation, it's very difficult to understand what the networks of genes that they're using might mean, because you're capturing probably around 12,000 transcripts for any group of cells. But when you have an atlas approach and you can compare large numbers of tissues against one another, very quickly you are able to see which types of transcripts are able to group cells with similar anatomical or developmental properties together. And patterns that may be anecdotal in an individual experiment become generalisable and even rules when you start looking them en masse like this. So these are incredibly valuable resources. The difficulty with transcriptional profiling, and again as Nathan's indicated, is that it's just one part of a layer of molecular information that contributes to how we describe the phenotype of a cell. DNA here is the easiest part of the information to gather because it's relatively invariant from cell to cell and actually relatively invariant from person to person, and actually relatively invariant from species to species, although there are clearly some caveats to that last point. The difficulty here is that we think we've got between 20 and 30,000 genes, but we actually only know what about 7,000 of these genes do. 7,000 genes which we've annotated to a pathway or a network reliably. And for the rest, we might have a name. That name might be associated with some kind of protein domain. It may be named after a tissue it's been discovered in, but we know very little about their functions. And an even greater proportion of protein-coding genes, an even greater proportion of genes are not protein-coding, I should say, but they are non-coding RNAs. And their role probably extends across this entire molecular network in terms of not just designating phenotype, but also stabilising that phenotype as well. Of course, as we move into those molecules which are produced in response to environmental stimuli, RNA, protein modifications, and of course the entire metabolite protein and metabolite genome interaction, then the level of complexity that we're dealing with also increases. And this is a reminder to us that cells are not static, that we can't take one individual RNA snapshot and really understand what the phenotype of that cell is. We need a way of integrating all of this information together and filtering it down to that subset of really relevant molecules that can tell us something about the phenotype that we're interested in. And then, of course, we need to remember that there is a level of variability in biological systems that's not just a reflection of a different way two people in a lab might pipette, or different genetic backgrounds that we all contribute to these kinds of biologies, but actually also represent a fundamental flux, a fundamental component of cell systems themselves. Because cells are responding to environmental cues and then returning to a stable state, because cells are dividing, we can think about these networks of genes and proteins and other molecules constraining the phenotype of a cell such that some cells are going to be deep down in this phenotypic well. The entropy for change is going to be very, very low. They're going to be resistant to moving from this state. Perhaps they don't express the receptors of the growth factors that are being challenged across them. Perhaps they're just about to enter, or they've just moving through the cell cycle, so most of their energy is going into cell division rather than differentiation. And in this stem cell population, we have just a very few number of cells which are not in the cell cycle G2 or M phases, but are expressing growth factor receptors, and where entropy for change is high, but our understanding of which way they're going to change is low. So networks don't just enable the phenotype, they also constrain the phenotype of a cell, and they buffer the phenotype of a cell from these extrinsic events. And I think some of the questions that we should be asking of these stem cells is really, how stable is the phenotype that we're measuring? How predictable is any direction of change as we differentiate a population of cells? And more importantly, what is the flux in the system and how do we use this to our advantage? If we took this network, this pluripotency network, and compared human, mouse and pig, we know we're going to see differences. Some of these are going to be because the cells may not be equivalent states. Some of them may be because different species simply use different networks. And Dee showed us this very beautifully last session. So we really need to understand that variability in this network is a pretty important factor, and we should be exploring it. This picture of Waddington's epigenetic landscape is in most biological textbooks, and I think most of you would be familiar with it. And if any of you are playing bingo, I guess now would be the time to hit one of the bingo buttons. But I often think that this particular diagram is misunderstood. And I really like Conrad Waddington's description of this landscape. So what he's describing is the canalisation of a cell through development. And originally he talked about it in terms of these bifurcations or decision points, at which there was really a point of no return. A cell moved down this landscape, it funneled down into one of the ruts, and then you ended up with a differentiated cell type. And we consider that the poles underpinning this landscape are the networks of genes, which might be describing the pluripotency network here, or the differentiated cell network here, and that these humps are really describing the epigenetic barriers which prevent a cell moving between these different regions. But of course, Waddington also talked about canalisation as allowing variability. In a canalised system, a cell can actually take a different route down any one of these valleys. What you're looking at is the path, the common path. Oh, that was good. What you're looking at here is the common path, but of course a cell could run down the edge and still reach the end here. So canalisation allows for a level of phenotypic variation, and I think this is important when we're thinking about expressivity of phenotypes, penetrance of genetic mutation, and when we're talking about including variability in our networks. So last year, the FANM group, which published the big transcriptional atlas, also looked at differentiation in a large number of cell types. Here across these x-axes you can see time measured in minutes, and they took a series of different cell types, including quite a large number of pluripotent cell types, and differentiated down different mesendoderm and neuroectoderm lineages. And they asked, what are the classes of transcriptional change that you see, And what you're seeing here is that the very, very first thing that happens, really within the first few hours of change, the first hour indeed, is a change in the enhancer landscape of the transcriptome. And then you see a change in the transcription factor genes, and then you see a change in the structural genes. So transition out of a pluripotency network requires first remodelling of the chromatin landscape, and then instruction of the new transcription factors for the next canalised network, and then the structural proteins, which are going to be the proteins which actually do the function of that new cell state. What about reprogramming? If we're taking a cell and we're moving it back up this barrier, and we know, thanks to Yamanaka, that all that is needed to reprogram a cell from a differentiated state back to a pluripotent state is the expression of 3 or 4 transcription factors. That's enough to reprogram. So Andras Nagy, as part of the Project Grandiose team, asked this question in mouse. He took a fibroblast from a mouse that had already been reprogrammed once. It had been reprogrammed with a piggyback transposon, which could be turned on and off with the presence of doxycycline. And when he measured reprogramming under a low level of doxycycline, he was able to recapitulate the normal induced pluripotent cell state, or this normal ES cell-like state. But when he exposed the cells to really high levels of doxycycline, he got this funny-looking cell. They called it the F class because it's a fuzzy-looking cell. And the fuzzy cells turned out, through the chimera assays, to be pluripotent. But they don't share the same transcriptome as an ES cell, and they don't share the same epigenome. In fact, these are quite synthetic cells. They don't last in the absence of this high dox. So you have to have very, very high levels of nanog in particular superimposed on these cells to maintain that cell state. So this also gave us some insight into the fact that we are often constrained when we talk about pluripotent stem cells to thinking about those cell types that we know from developmental or anatomical niches. But actually, there are opportunities with these stem cells to be exploring landscapes well outside our what's known. Combinations of networks and genes in networks which haven't been seen before and which can give rise to completely new cell states. And this era of synthetic cell biology, I think, is very exciting for its potential. The other thing that was discovered in this Project Grandiose program was that, and I think I've got a slide for it here, the very first changes that happen as you move a fibroblast back up that Waddington landscape to a pluripotent-like cell is the small RNA and transcriptome changes first. In fact, it changes very rapidly. But the epigenome, and in particular DNA methylation, this is the last thing to change. And so what we've learned from this is that one of the roles of permanent chromatin changes, such as seen with mitotically heritable epigenome changes, is that this stabilises cell phenotype rather than driving cell phenotype. So we often think about the genome as a book or a map, but I'd like you to think about it more as a library. It's a library with almost infinite numbers of stories to be told. And then, if we consider that, then this idea that a stem cell has got every shelf, every possible story available to it doesn't really make sense. In fact, there's a separate stack just for the pluripotent stem cells, and different pluripotent stem cell types might be reading a different storybook. And to get this stem cell through to a differentiated cell type, it actually has to transition through different cell states. It's very hard to get it to directly go to a mature cells. And these epigenetic factors, like librarians, their roles are to organise the stacks and just to bring the books that the cell needs to the front of the library. So their role is to stabilise, not to direct state change. In the very last part, I want to point out to you that that moving through all of these layers of data can be very confusing, and for non-bioinformaticians or people who, unlike me, don't like looking at sequence, you know, it can be very hard to know how to get traction onto these. So systems biology has got some fairly rigorous principles to move through different types of data. You've got this idea of emergence, which is the idea that combining everything, you get something bigger in return inside. Modularity, this idea that proteins work in networks and pathways. And this idea of robustness, which means that you can have a level of phenotypic variation while still belonging to the same phenotypic class. And so in order to navigate this, we need not just to generate lots of good quality data, we need tools for looking at it. And I really just want to throw at you a few screenshots of this platform that we've built called Stemformatics. It's a free resource, it's available to all of you. It's got lots and lots of interesting information in early embryogenesis, from primary embryogenesis and directed stem cell differentiation. It's easy to search, Google-like terms. It's got ways of comparing the expression of different genes. Here's beta-actin, it's expressed pretty ubiquitously. Here's OCT4, it's really expressed a whole lot in pluripotent cells and not in anything else. Here is DMT3L, which is really restricted to very early embryogenesis, the early blastocyst stage in humans here. You can look for your favourite gene. In this case, I'm looking for Minkle, which is my favourite gene, and you can see that your gene may be expressed very highly in a certain cell type. Here, Minkle's really highly expressed in innate immune cells and not at all pluripotent cells. And you can dig in and have a look at the individual data sets that have contributed to this profile. So it's really handy if you just want to know whether the gene you're interested in is really that unique to a particular cell state, or whether it's reproducibly seen in the cell state you're interested in. You can build heat maps if you've got gene lists that you're interested in looking at. And that's it, really just to acknowledge that I talked about the Phantom work, which is a very large program run by Riken Japan under the guidance of Yoshihide Hayashizaki, Piero Konichi and Al Forrest. And I talked about Project Grandiose, which was the brainchild of Andres Nagy in Canada, but had a number of Australian contributors. Thank you.